Welcome to your Carbohydrate Metabolism podcast, where you're going to learn more than you've ever wanted to know about the metabolism of carbohydrates. Let's begin. First and foremost, I need you to not pause me, and please do not fill in all this information. What I need for you is to print off your blank version of this, which can be found on Canvas. So print that off. You should not be copying this down on the slide. I repeat, do not copy this down right now. We are going to walk step by step through this, and as we go, we will fill in our blank little graphic organizer, and we'll talk about each of the different steps. We'll color code them, but for now, I just want you to see that forest level view of here is where we're going, and this will all make sense, hopefully, by the end of this podcast. But don't panic yet. I will tell you if you need to panic, and it's not yet. The second graphic organizer that we're going to be referencing throughout the next 3 podcasts is this one. So you have a blank version of this as well. Again, don't copy this down yet. This doesn't make any sense yet. We're going to use this as a way to orient ourselves for protein, carbohydrate, and fat digestion. So for this podcast, we are only focusing on the digestions of carbohydrates. So we're going to talk about what we start with, where we go, where we get our energy from. You will see digestion of fats another place. You'll see digestion of proteins in another podcast. So hang on to that. This is just kind of a way to make sense of all the information that you're going to be getting. So as we go through this, some of this is going to be super obvious to you. You're going to be like, McCann, I learned this when I was like 5 years old. We're going to get real complex real quick, but let's just start with the basics, shall we? So, as a lot of you know from previous courses, carbohydrate metabolism begins in the mouth. You have an enzyme called salivary amylase. Again, notice that ASE ending that starts to break down polysaccharides into monosaccharides. And a good example of this, if you're feeling incredibly bored during this quarantine, is to take some saltine crackers or any type of cracker and chew them. Don't swallow, just chew and chew and chew. After about the minute mark, you're going to notice that those saltine crackers start to taste sweet. So what you've done is you've broken down the polysaccharide into individual monosaccharides or glucose molecules. And your salivary amylase is the first step in carbohydrate digestion, and that starts in the mouth. It's kind of a gross experiment, so if you gag easy, please don't do this. Partially digested starches then enter the stomach where it's acidic, and digestion continues into the small intestine. It's broken down into monosaccharides there. As it moves through the small into the large intestine, nutrients are absorbed out of those carbohydrates through the intestinal wall and eventually move into the bloodstream. So now is time you can start filling in your graphic organizers. So we're going to start with our big overview of the 3 different types of digestion. Starting with digestion of carbohydrates. So carbohydrates are going to be first broken down into monosaccharides. So we start with polysaccharides, they get broken into monosaccharides. Those individual monosaccharides, and we're going to talk a lot about glucose specifically, go through a process called glycolysis, and this is where our other graphic organizer is going to come into play. This is essentially looking at glycolysis and the citric acid cycle and oxidative phosphorylation. So that is essentially, this portion will move to a different graphic organizer. But your glucose will be converted into other intermediate products that can enter your citric acid cycle, where we're releasing these high energy electrons so that we can do work and produce ATP. The whole goal of all of these metabolism is to produce ATP, which is the currency for energy for your cells. So all cells use ATP to do work, and this is how we free that ATP from food sources. Okay, so thinking back to 1201, carbohydrate metabolism yields 4 kilocalories of energy per gram. That was the unit conversion that we used in our nutrition section about this time last semester in 1201. So the first step of carbohydrate metabolism is called glycolysis. So you need to know the first step is glycolysis, and this produces some energy from carbohydrates. Glycolysis produces 2 ATP, and that's a net amount. It actually costs 2 ATP for glycolysis to work, and we get 4. So the net is 2 ATP that we get out of this process, and you also get NADH. NADH is, think of it like an electron carrier. So it captures electrons from the breakdown of this monosaccharide glucose, and it carries it to another part of the cell where it can be used to create ATP. Glucose is converted into pyruvate and these electrons. So real quick, let's talk about this. So you have an electron carrier, NAD+, and that is converted into NADH. So NAD+ picks up 2 electrons. So if we were just to combine these two together, we start with a +1 charge. Sorry, let me fix this. So NAD +1, and if we add 2 negative charges, we would have NAD-. So NAD+ picks up 2 high energy electrons, and it also picks up 1 proton, and we'll see why this proton is important later because we're going to use it to also create ATP later on. So when we pick up that proton, that has a +1 charge and it has a hydrogen, and that's how we wind up with NADH. So NAD+ is reduced, meaning it gains electrons, and so this is its reduced form. The process of glycolysis is anaerobic, which means you don't need oxygen for glucose to go through this first step of carbohydrate metabolism, and it happens in the cytoplasm. So you're probably asking yourself, this is a lot of information. What am I going to be responsible for knowing come test time or come just general knowledge I take with me out of college? You need to know the name of the process. You need to know the location it happens in. So this is glycolysis. It happens in the cytoplasm. You need to know what it needs to start. So you need the reactants, in this case, a molecule of glucose. And you need to know what the products are. I produce pyruvate, high energy electrons, and NADH. All right, guys, it's not time to freak out yet. It's time to fill in our graphic organizer. So follow along with me as we go. So I am going to color code. I'm going to use the highlight function to highlight the steps of glycolysis, and I'm going to highlight those in green. So glycolysis is going to be green on here. You can color code if that makes sense to you. If it doesn't, don't worry about it. So glycolysis happens in the cytoplasm. Okay, so we are in the cytoplasm of the cell. We're not in the mitochondria, which is the powerhouse of the cell. It's where the energy is, I don't like the word produced, but it's converted. Okay, so glycolysis is happening in anaerobic conditions in the cytoplasm. It takes 2 ATP to start. That's called an energy investment. And it also takes 1 molecule of glucose. So I need a molecule of glucose, I need 2 ATP to get glycolysis going. Glycolysis will form 2 pyruvate, so that's a product. Okay, and it also produces 4 ATP or 2 ATP net. Okay, so it produces 2 overall from the process. What is important also is that it produces these electron carriers. It produces this NADH. Notice that the NADH is going to be used in a step later on. We'll talk about how this is used later. But for now, just know that those 2 NADH are going to go to the inner mitochondrial membrane, where they're going to be used. So these 2 pyruvate are going to go through another reaction so that they can enter into the citric acid cycle, but we're not ready for that quite yet. So what we just talked about can be summed up by these green highlighted areas. That is glycolysis. Start with 2 ATP and a glucose, you form 2 pyruvate, 2 ATP, and NADH. All right, so we have our 2 pyruvate now. And depending on the conditions, its fate is going to be different. So if there is no oxygen, your pyruvate is going to undergo fermentation. If there is oxygen, it's going to proceed to the acetyl, or it will be converted to acetyl-CoA and enter the citric acid cycle. So under anaerobic conditions, your cell, and I say you, I don't really mean you, but under anaerobic conditions, human cells will undergo lactic acid fermentation. That happens in the cytoplasm. If you're a yeast, you'll undergo alcoholic fermentation. So if there's no oxygen and you have a yeast cell, the glycolysis the pyruvate will enter into alcoholic fermentation and you'll produce CO2 and ethanol. Thankfully, humans don't go through that, or every single time that we run and we don't have any oxygen left in our cells, we would produce ethanol and get drunk. Lactic acid fermentation is what we go through. So we produce lactic acid in the absence of oxygen from our pyruvate. If conditions are right and we have oxygen in our cells, your pyruvate is converted into acetyl-CoA and it will enter into the mitochondria where we can produce loads more energy for the cell. Going back to our graphic organizer, let's look at the difference between conditions. So if I have anaerobic conditions, my 2-pyruvate, and I know we've already highlighted this as being a product of the green, but it's also a reactant for fermentation or aerobic respiration. So the 2-pyruvate under anaerobic conditions will form 2-lactate or lactic acid and 2-NAD+. So this is the oxidized form of NAD and it can pick up electrons again. So it kind of is a stopgap measure so that we can keep processing glucose and the organism doesn't die. This won't work forever because it doesn't produce enough ATP for the organism to live off of, but it will work, in case you have a moment where you have depleted the oxygen in your cells and you're waiting for more oxygen, like when you're exercising. Under aerobic conditions, your 2-pyruvate are going to form acetyl-CoA. We will talk about that in the next slide and we'll fill that in just a moment. So this whole section right here is showing us if there wasn't any oxygen, we would go through fermentation. All right, so oxygen is present in the cell. My 2-pyruvate get converted into acetyl-CoA, and that acetyl-CoA can now proceed to the Krebs cycle. So you will see some people who call this the Krebs cycle. That's kind of the old school name for it, named after the scientist who discovered it, Hans Krebs. Nowadays, though, in your textbook, you're going to see it labeled as the citric acid cycle, or CAC. In this reaction, when you go through the citric acid cycle, you're producing 2 ATP. So we're still not producing a ton of ATP through this. What's more important to this stage is that we're releasing electrons, which are again picked up. This time we're picking them up by NAD+ and FAD. This happens in the mitochondria. So if there's oxygen, we can proceed to the mitochondria. And I know we already talked about NADH, but our FAD, we would add 2 electrons to this. So it's neutral to begin with. Here's 2 negative charges. We're going to have to add 2 protons or 2 hydrogen ions in order to get a neutral form. So just wanted to give you an idea of where those 2 H's came from versus the 1 H that's picked up in NAD+. So those are going to travel to the electron transport chain at the very end, and we're going to use those protons, we're going to use those high energy electrons to do work for us to produce ATP, but not yet. So the goal of this is to produce these electrons that are carried to a different location, and we produce a little bit of ATP in the process. All right, we got a new step. Let's fill in our graphic organizer. So at this point, our 2-pyruvate have been converted into 2-acetyl-CoA. So the 2-pyruvate under aerobic conditions are converted to the 2-acetyl-CoA, and the acetyl-CoA can enter the mitochondria. And this is what will enter your citric acid cycle. So as a result of your citric acid cycle, you're going to wind up with high energy electrons and protons, and these are carried over to the inner mitochondrial matrix. That's where they're going to do work for us and produce the majority of our ATP that we get through aerobic respiration. In the process, you'll produce 2 ATP. So far, just to note, we have produced 2 ATP through glycolysis, and we produce 2 ATP through the citric acid cycle. Something else to note is that 1 glucose produces 2 pyruvate. 2 pyruvate are converted to 2-acetyl-CoA, but the citric acid cycle can only take 1 acetyl-CoA at a time. So 1 acetyl-CoA goes through the citric acid cycle, and we reform our product, and then the second one goes through. And there are a lot of intermediate steps that I don't need you to know. I need you to be able to see the big picture of this. So for each step, what's the name? What does it need to start? What does it produce? Where is it happening? Remember when I told you not to freak out? Still not time yet. I will tell you, it is not time to freak out. I just wanted to give you a little bit of an idea of how much more complex the citric acid cycle is. So our pyruvate is a 3-carbon molecule, and that is going to be converted into acetyl-CoA. We lose a molecule of CO2 in the process. So we often talk about metabolism and cellular respiration as being glucose in the presence of oxygen breaking into carbon dioxide and water. It's a combustion reaction. Except for that we can't combust all of those and break all of those bonds at once, or we'd go up in a big ball of flames. So this is the process that we do it very slowly in a very controlled manner and release just a little energy at a time so that we don't go up in a big ball of flames. So acetyl-CoA enters the citric acid cycle. We release molecules of CO2. Notice here we start with NAD+, it gains electrons or it's reduced, and it forms NADH. Those will be shuttled off to the inner mitochondrial membrane to the electron transport chain. We also at the same time will phosphorylate an ADP molecule, and that's the low energy form, storing energy as ATP. So think of this as being the form of ATP that we can use to do work. FAD will pick up 2 high energy electrons as well as 2 protons, and that will be shuttled off to the electron transport chain in the inner mitochondrial matrix as well. We reform our starting products, and now we're ready for the second acetyl-CoA that was formed. So we actually have 2 turns of the citric acid cycle. Again, you don't have to memorize this. This was just kind of an FYI, there's a little more going on. So the last step of this is your NADH and your FADH2 are shuttling electrons and protons to the electron transport chain, so hydrogen ions as well as your electrons. This is where oxidative phosphorylation is going to happen, and this is our moneymaker when it comes to ATP. We are going to form 32 to 34 ATP. And we are using electrons that were released through previous stages when we broke down glucose to pyruvate and when we broke down our acetyl-CoA. And we released electrons at each of those steps. Now you're going to see what we do with those electrons and what we do with those hydrogen ions. Okay, so this final stage is our electron transport chain, ETC, and our oxidative phosphorylation. This is where we get all the electrons from, or all the ATP from. So throughout various processes, we have these electron carriers shuttling electrons and hydrogen ions over to the inner mitochondrial membrane. Okay, so glycolysis gave us some. What else gave us some? A citric acid cycle gave us some. Now what are we going to do with them? Those high energy electrons are being dropped off in the electron transport chain, and that's what you're seeing right here. This is the electron transport chain, and it's a series of proteins that are embedded in the inner mitochondrial membrane. So as the electrons move from one protein to another, they lose a little bit of energy. That energy is being used to drive proton pumps. Okay, so we drop off our hydrogen ions, and normally your cells would want to have an isotonic solution, or they would want to have equal numbers of protons on one side of the membrane versus the other. Okay, usually it doesn't take any energy, whichever side has the higher concentration. You will see a movement from high concentration to low concentration until both sides are equal. But in this case, we need to create a higher concentration of these hydrogen ions on one side of the membrane versus the other. And to do that, we're going to have to pump them through these protein channels. And in order to pump them against their concentration gradient, it's going to take energy. We get that energy from those high energy electrons that got dropped off in this electron transport chain. So the electrons are powering these protein pumps that are moving hydrogen ions against their concentration gradient. So see how many of these hydrogen ions we have on one side of this membrane. Well, as I said before, those hydrogen ions want to go back and have equal concentrations on both sides of the membrane. There is one location that they can go back across the membrane without any energy required, and that is through this little molecule called an ATP synthase. Notice the ending. It's an enzyme. So your hydrogen ions are moving back across the membrane with their concentration gradient from high to low, doesn't take any energy. As they move through this, those hydrogen ions are powering an ATP synthase. So you would put in 3 hydrogen ions, that gives you 1 turn or 1 crank of your ATP synthase. And what that does is it phosphorylates an ADP and an inorganic phosphate to form an ATP. So this is how we make ATP. ATP synthase phosphorylates that. And so when you think of ATP synthase, think of those old-timey souvenir machines that you would see, God, you see them at the zoo still, where you have to put in like 3 quarters and a penny, and then you turn the crank and it keeps your 3 quarters, but it spits out that flattened penny that's that souvenir from whatever location you have. That's kind of like ATP synthase, where you have to put in a certain number of hydrogen and you get a phosphorylated ADP out of it. So you get ATP out of it. Whew. Okay, still not time to freak out yet. Just want to point out all of the hard work that you guys have done. We have made 2 ATP. You've already got this color coded. Don't worry about a new color. From glycolysis. That's our net. You have released 2 ATP from the citric acid cycle. Oxidative phosphorylation, in the presence of oxygen, this is your moneymaker. You have released 32 to 34 ATP from 1 molecule of glucose through the electron transport chain and oxidative phosphorylation. So if you were to add all these together, 1 molecule of glucose will give you, or will yield, 36 to 38 ATP. This is not a steadfast rule. Depending on what type of cell it is or how efficient it's running at the time, this number is variable, but on average you get about 36 to 38 ATP per molecule of glucose, and they're coming from each step. So notice my anaerobic steps doesn't yield very much ATP. Citric acid cycle only happens in the presence of ATP, yields 2 ATP, that's not very much. But what we really want is a presence of oxygen in the mitochondria, and it's this proton pump, okay, this hydrogen ion pump in the electron transport chain that's driven by the electrons that are released from all the prior steps of breakdown of glucose, that's giving us the most ATP. And that's the real moneymaker for the cell, and that only happens in the mitochondria, which is why it's considered the powerhouse of the cell. All right, so again, to sum up what point I just made, 1 molecule of glucose is going to yield about 36 to 38 ATP. Your book breaks it down just a little bit differently, but for our purposes, 36 to 38 will work. All right, this lovely young woman has just run and fatigued her muscles and her cells. When you do vigorous exercise, that's going to deplete your cells of oxygen. If you deplete your cells of oxygen, when you're breaking down your glucose to 2-pyruvate through glycolysis, you can assume that there's no oxygen. You're going to force yourself into anaerobic conditions where you're starting to produce lactic acid. So a buildup in lactic acid in the muscles is going to cause soreness and cramping because you've created an oxygen debt. When you take in deep breaths of air after exercising, you're going to repay the oxygen debt, and your lactate will slowly and gradually reoxidize to pyruvate. So that was this little box that we kind of ignored before. So our lactate can reoxidize to pyruvate, and if there's oxygen, they can then enter into the mitochondria, get converted into acetyl-CoA, and enter the citric acid cycle. But until you repay the oxygen debt, it's going to be soreness for you. All right, so if you have any tissues deprived of oxygen, your pyruvate is going to be converted to lactic acid or lactate, and you're going to have elevated levels of lactate. We can use a blood test and determine if you have elevated levels of lactate as a diagnostic tool. So for example, if you had a heart attack or some sort of blockage in one of your coronary arteries, so blood's not flowing to this tissue, the oxygen supply is therefore cut off, and we would expect our muscles to start to die. In the lack of oxygen, we're going to see an increase in fermentation and an increase in lactic acid production because that muscle is blocked. So seeing an increase in lactic acid is one indication that your patient may have or may be having a heart attack. It's also used to assess how severely ill an individual is. So having a higher than normal concentration of lactic acid can indicate that you have lung disease, congestive heart failure, or the presence of a serious infection. So it doesn't necessarily mean you're having a heart attack. It could be the presence of something else, but it's a great first step in diagnosis. Fermentation isn't all bad, guys. Fermentation is the reason that we have all the foods that we love. Fermentation plays a key role in the production of bread, and I don't see cheese on this plate, but cheese too. There's cheese. Yum! And it also plays a role in the production of beer. So alcoholic fermentation or ethanol fermentation will produce beer, whereas lactic acid fermentation produces cheese. And we use fermentation to produce the bubbles that you see in bread, and we bake off the alcohol that is produced in the dough. Could you eat the dough and get drunk? No, but you're going to get a stomach ache, so don't try. Yeah, fermentation, not all bad. I forgot that I never told you when it was time to freak out, and we're finally done with this very, very long podcast. So if you need to freak out, you have my permission to freak out, but you made it, you survived.